The Gram stain, the most widely used staining procedure in bacteriology, is notable because it distinguishes between cells with different cell walls. Gram-positive bacteria have thick cell walls that contain at least 3 layers of peptidoglycan. These stain purple. Gram-negative bacteria have 1 layer of peptidoglycan and a high lipid content. These stain pink. In this video, we'll go through the steps of the Gram stain and how it distinguishes between types of cell walls. Then, we'll talk a little bit about another type of cell wall called the acid-fast. By the end of this video, you should be able to explain the medical significance of mycolic acid and give examples of bacteria that contain it. You should be able to explain the steps of the Gram stain and explain how energy production is different between prokaryotes and eukaryotes. As you recall, the cell wall of most bacteria, probably over 90% of them, contains a disaccharide called peptidoglycan. The disaccharide alternates forming a backbone, and the backbones are connected to each other through chemical crosslinks. Remember that the chemical crosslinks come through one of the sugars of the disaccharide called N-acetylmuramic acid, or NAM for short. NAM is exactly like its counterpart, NAG, which is N-acetylglucosamine, except for the fact that it has an amino acid tail, which you can see here. Chemical bonding between the amino acids of one tail and the amino acids of another tail tie the backbones of peptidoglycan together in the cell wall, strengthening it. We also said in our last video that your cell walls are classified into 2 categories. You have the gram-positive, which has at least 3 layers of peptidoglycan. So these blue boxes here, each one represents a layer of peptidoglycan. All of these are cross-linked one to another, and they're kind of bound together by the cross-links and teichoic acids, which are embedded among the layers of peptidoglycan. Gram-negative cell walls, on the other hand, have 1 single layer of peptidoglycan, and it's not cross-linked. Remember, it's actually connected to the outer membrane of gram-negative cells via a special lipoprotein, which we called bronze lipoprotein. The outer membrane is extremely protective for your gram-negative cells. It excludes certain antibiotics and disinfectants from the cell. So, the gram-positive cell wall with the multiple layers of peptidoglycan stains purple during a Gram stain. The gram-negative cell wall, which only has 1 layer of peptidoglycan, stains pink or red. Why do we need to stain bacteria? We stain bacteria because they are colorless. If we don't add color to these cells, they'll be very difficult to see. During a Gram stain, you take bacteria and you apply them to a glass slide. Now remember, the bacteria are colorless at this point. You allow the bacteria to dry, and then you pass them through the flame of a Bunsen burner. The heat from the Bunsen burner denatures proteins on the surface of the bacterium, and this causes the bacteria to stick to the glass slide. This is important because if we don't do this step, when we wash the slide with several chemicals in order to color the bacteria, they would wash off. So we heat fix the bacteria to the slide so that as we pass all these chemicals over the slide, the bacteria stick a little bit better. So these are the chemicals that we use. The first one is called Gram's crystal violet. And this chemical is going to penetrate the cell wall, and it'll go into the cytoplasm where it stains components in the cytoplasm as well as in the cell wall. Because this is a violet colored stain and it penetrates so well, all cells, no matter whether they're gram-positive or gram-negative, stain purple. Next, we add another chemical called Gram's iodine. Gram's iodine is what is called a mordant. Mordants increase the ability of the first stain to stick to the cells. In this case, the iodine binds the particles of crystal violet, making them a bit larger and harder to wash out of the cell. But at the end of this step, all cells are still purple. Next, we rinse the slide with water, and then we're going to do what's called a decolorizing step. This is the step that's going to distinguish gram-positive cells from gram-negative cells, because when we add this decolorizer to the cells, one set of cells will stay purple, the other one is going to be decolorized. So our decolorizer is 95% ethanol. And when we add this alcohol, this ethanol, to the cells, it causes the peptidoglycan in the cell wall to dehydrate. So we're going to remove water from it. So if you think about a sponge that you have on the sink, when it's wet, the holes are nice and big, right? But when the sponge dries out, what happens to those holes? They get smaller. Same thing happens with peptidoglycan, and this is going to trap the Gram's crystal violet / iodine particles inside the cell. So the cell stays purple because those iodine crystal violet particles are trapped. For the gram-negative cells, on the other hand, remember we only have one layer of peptidoglycan. So when that layer dehydrates, the peptidoglycan develops rips and holes in it. The other thing that happens is that outer membrane gets washed away because alcohol is able to solubilize lipids. So now that we've removed the outer membrane and we've made rips in the peptidoglycan layer, the crystal violet iodine particles are easy to wash out. So these cells lose their purple color. Now we've got a problem. We have some cells that are stained, those are the gram-positive, but we have some cells that are not stained. They've been decolorized, those are the gram-negatives. So in the last step of the gram stain, we add another dye called safranin. Safranin is a reddish-pink dye, so when we add this to both cells, the gram-positive cells don't take up the safranin because they're full of Gram's crystal violet. So those cells still stay purple. The decolorized cells, on the other hand, will take up the safranin, and so they'll stain pink or red. You'll need to be able to describe this procedure for your exam. Usually it's an essay question, so you might want to practice writing out the steps of the gram stain and the colors that you expect, so that when you get to the exam, you know how to answer this question by heart. So now here's a way to remember some of the basic characteristics of your gram-positive cell. This is sung to the tune of Happy Birthday. Hopefully you know this song. I want you to sing it with me, and then we're going to go into the lyrics for the gram-positive cell. Here we go. Sing with me. Happy birthday to you. Happy birthday to you. Happy birthday, Bacillus anthracis. Happy birthday to you. Gram-positive cell stains purple swell. Lots of pepto, thick cell wall, tycho as well. Gram-positive cell stains purple swell. Lysozyme kills it, penicillin as well. All right, I know that was embarrassing, but hopefully that's going to get caught in your brain and you'll remember the following characteristics of gram-positive cells. First thing you need to remember, gram-positive cells stain purple with the gram stain. And the reason for this is they have at least 3 layers of peptidoglycan in their cell wall. At least 3. So we say that these cells have a thick cell wall as a result. Embedded within the peptidoglycan, we have those teichoic acids. Remember, they help hold the cell wall together. They also provide a few other functions, but we're not going to worry about those for this class. Myco here refers to mycolic acid. We're going to get to that in a couple of slides, but for right now, mycolic acid is embedded within a thick type of cell wall. So it would be like a gram-positive cell if the mycolic acid wasn't there. Lysozyme is an enzyme that's in your sweat and in your tears. It breaks the bond between NAG and NAM in the peptidoglycan backbone. This significantly weakens the cell wall, and it makes these types of cells susceptible to osmotic lysis. Penicillin is a drug that prevents the cross-linking of the NAM molecules, and it also stimulates the production of autolysins, a group of enzymes that actually break down the cell wall as part of normal cellular life. So it's like a 1-2 punch of death with this particular antibiotic. This antibiotic is effective only against your gram-positive cells. So we can't do gram-positive without doing gram-negative. This song is to the tune of If You're Happy and You Know It. So I'm going to sing it. I want you to sing it with me. Don't leave me hanging out there. So we're going to sing this together, and then we're going to move into the lyrics for this cell wall. If you're happy and you know it, clap your hands. If you're happy and you know it, clap your hands. If you're happy and you know it, then your life will truly show it. If you're happy and you know it, clap your hands. If the cell has lipid A, then gram-neg. And 1 layer peptidoglycan, look gram-neg. If the cell is staining pink, and then you ought to start thinking outer membrane hard to kill, then gram-neg. So, remember, one of the big characteristics of your gram-negative cells is that they have that outer membrane. And embedded in the outer membrane is that lipopolysaccharide. At the base of the lipopolysaccharide is lipid A. And remember, this is an endotoxin. It causes physiological responses like fever and drops in blood pressure and vascular collapse and things like that. Also, the outer membrane has that lipopolysaccharide that I've already mentioned, and remember the negative charge of the lipopolysaccharide repels certain types of disinfectant. Antibiotics also have a hard time penetrating these cells because of that outer membrane. The phospholipid bilayer makes a barrier that most antibiotics can't diffuse through. They can't penetrate it. The only way that antibiotics can get into the cell is via porin proteins. So if certain antibiotics like penicillin are too large, they're excluded from the cell. This makes the cells hard to kill. Okay, they're hard to kill because of the outer membrane and the lipopolysaccharides that are embedded within it. Remember that in addition to the outer membrane, these cells only have 1 layer of peptidoglycan. And it's because of the outer membrane and this 1 layer of peptidoglycan that when we add alcohol to these cells, they decolorize. Then we add safranin, a reddish pink dye, and that causes these cells to stain pink. So, 2 songs, keep them in mind. They kind of summarize big ideas for both types of cell wall. But those aren't the only types of cell walls. We also have another type of cell wall called the acid-fast cell wall. And this cell wall has a thick, waxy lipid in it called mycolic acid. It's embedded among the peptidoglycan. So think about when you wax a car. What happens when it rains after you've freshly waxed a car? The water beads up and it rolls off, right? It's the same with this type of cell wall. Disinfectants, antiseptics, Certain other types of chemicals and antibiotics can't penetrate that waxy layer. They just beat up and roll off, which allows these cells to survive. In addition, when these cells are phagocytized, phagocytosis is harder because of the mycolic acid, and it's also hard for phagocytic cells to digest these bacteria. So for example, bacteria belonging to the genus Mycobacterium will have mycolic acid in their cell wall. So Mycobacterium tuberculosis, the bacterium that causes tuberculosis, is kind of famous because it's able to live inside of macrophages, these phagocytic cells that help defend our bodies against invading cells. Well, since they can have this mycolic acid, they survive inside of these macrophages. That makes it very hard to cure tuberculosis. Mycolic acid also makes it difficult to stain cells, so we usually have to use a stain like carbofuchsin that contains a chemical in it that has kind of a lipid base so that it can actually help the stain to penetrate that mycolic acid layer. Cells that have mycolic acid, like these M. lepore cells here, that's Mycobacterium lepore, by the way. These M. lepore cells stain pink because of the carbofuchsin. They actually wind up retaining that in the mycolic acid. And then cells that don't have mycolic acid, like these Staph aureus cells here, they'll stain blue. We call cells that do have mycolic acid acid-fast, and cells that don't have mycolic acid non-acid-fast. So not all prokaryotes actually have a cell wall or have a peptidoglycan cell wall. Mycoplasmas are bacteria that actually don't have a cell wall. They're the smallest known cells, certainly the smallest known bacteria, and they, instead of having a cell wall, have sterols in their plasma membrane. Sterols are steroid alcohols like cholesterol, so they're kind of rigid molecules and they give some stability to the plasma membrane. The only reason that mycoplasmas don't burst from osmotic lysis is because they're so small. Archaea are also prokaryotes. Some of them don't have a cell wall. Others have a cell wall, but it's made up of pseudomurine. So this is kind of like peptidoglycan, but it doesn't have the NAM molecules and the amino acids that are attached to NAM. So this information here goes back to lysozyme and penicillin. I've already talked about it, so I'm going to skip it for right now. If we treat gram-positive and gram-negative cells with certain chemicals, we can actually cause them to lose their cell wall. A gram-positive cell that loses its cell wall is called protoplast. Gram-negative cell that loses its cell wall is called a spheroplast. And both of these are susceptible to osmotic lysis because they no longer have that peptidoglycan layer, that rigid layer that helps keep them from bursting when their volumes increase due to osmosis. L forms are another type of cell that don't have a cell wall, and when they swell, they don't swell into just larger versions of themselves, but rather they swell into irregular shapes. The last thing I want to tackle really quickly is some differences in the plasma membrane and ATP production between prokaryotes and eukaryotes. First of all, the main function of the plasma membrane, regardless of whether you're talking about prokaryotes or eukaryotes, is that it confers selective permeability. It allows some molecules to come into the cell while excluding others. And the same is true for molecules leaving the cell. It allows some things to go out, but not everything. Now, a difference between your eukaryotes and your prokaryotes is, in addition to this function, In your prokaryotes, there will be enzymes for ATP production. So remember talking about something called the electron transport chain. It makes like the lion's share of ATP for eukaryotes like us, and it's located in mitochondria. Well, prokaryotes don't have mitochondria. So instead, their electron transport chain is actually in the plasma membrane of the cell. Another difference is you do have some photosynthetic bacteria like the cyanobacteria. The photosynthetic pigments that are responsible for this characteristic are actually located in folds in the plasma membrane that are called chromatophores or thylakoids. So those cells will appear green because of these little pouches containing chlorophyll and other photosynthetic pigments. This is different from eukaryotes. Remember, eukaryotes have chloroplasts that carry their photosynthetic pigments. So these little pouches that you see on the periphery of this bacterium are the chromatophores or thylakoids. These are the pouches that hold the photosynthetic pigments. Here at the plasma membrane on this side is where you would find the ATP synthesis proteins kind of embedded in there. They're so tiny you won't see them in this picture, but you just have to remember that they're there. Let's do a check-in to see what you remember. Which of these is a virulence factor? Is that the glycocalyx, mycolic acid, or gram-negative cell wall? There's more than one answer here, by the way. All of them. Remember, virulence factors are going to be those characteristics, those structures that increase the ability of a bacterium to cause infection and disease. The glycocalyx and mycolic acid definitely do that because they interrupt the ability of phagocytic cells to phagocytize certain types of bacteria. The gram-negative cell wall is definitely a virulence factor because it gives resistance to disinfectants, antiseptics, and certain antibiotics to bacteria. What kind of cell would this wall belong to? Would that be gram-negative, spheroplast, or gram-positive? Right, definitely gram-positive. You can tell because there are at least 3 layers of peptidoglycan. There's 1, there's 2, here's 3, and then you can see the teichoic acids that are embedded within it. Which of these is a characteristic of this cell wall? Does this cell wall contain an endotoxin? Is it sensitive to penicillin, or is it resistant to lysozyme? And I think there's more than one answer here. Right. This is a gram-negative cell wall, and it contains the lipopolysaccharide, which has lipid A. So it has an endotoxin. It's also resistant to lysozyme because of the phospholipid bilayer, which is colored yellow here. Good. Which of these is not true of the gram-positive cell wall? Is it that it contains lipoteichoic acids, it has multiple layers of peptidoglycan, or it stains pink during a gram stain? Which is not true? Of course. It stains purple during a gram stain, not pink, so that can't be right. Which of these is not part of peptidoglycan? Is it NAG, the outer membrane, or amino acids? The outer membrane. That's correct. Good job. So we're at the end of this video now, and I said that you should be able to explain the medical significance of mycolic acid. Remember, mycolic acid is a thick, waxy lipid that's in the cell wall of certain bacteria, and this is a virulence factor. It makes those bacteria hard to phagocytize and digest. we have a diagnostic stain that detects the presence of mycolic acid called the acid-fast stain. And certain medically relevant bacteria like Mycobacterium tuberculosis and Mycobacterium leprae have this mycolic acid in them. So they should stain pink, and we would call them acid-fast. I also went through the steps of the Gram stain and explained how those steps make gram-positive cells appear purple and gram-negative cells appear pink. You'll need to be able to explain that on your first exam. Finally, I talked about how energy production is different between your prokaryotes and eukaryotes. Remember, I mentioned that the proteins that are part of the electron transport chain are actually embedded in the plasma membrane of your prokaryotic cells. They aren't located in mitochondria because prokaryotes don't have mitochondria like eukaryotes do. I also explained that your photosynthetic pigments that are found in certain bacteria like the cyanobacteria are actually packaged in these little sacs or pouches that are associated or formed by the plasma membrane as well. So photosynthetic prokaryotes don't have chloroplasts like the eukaryotes do. So that means that the photosynthesis happens inside of those pouches, and that's a big difference. I hope this video was helpful. In the next video, we're going to wrap up our discussion of how prokaryotic cells kind of work and how they're different from eukaryotic cells. If you have any questions about this video, please feel free to write by email or text, and I'd be happy to explain more. I'll see you at the next video.